Okay, again, thanks for the opportunity to speak here. I'll talk to you today about cell-to-cell RNA transfer. So it's not quite axonal transport, it's transport of something into the axon from another cell. Here's one of your standard neuronal diagrams, and I just put that up to remind myself that the question we're trying to address here is, are all axonal proteins and RNA synthesized in the cell body? We're only talking about a tiny fraction of them, and the other aspect of this that we don't know about yet, in deference to Scott, we don't know whether growth and injury are special cases in which things are not synthesized in the cell body. So if you look at this diagram, is there a pointer? I guess I have one too. If you look at this diagram, you think about packaging everything from in the cell body to get down the axon. There's a shorter path. What if you made transcripts? What if you made proteins in the Schwann cells? This is a neuron from the penis. And what if you did cell-to-cell transfer into the axon itself? So that's what I'm going to talk to you today about, especially the case of RNA.
Okay, use that.
So our understanding of axonal and acceptance of axonal protein synthesis lags far behind our understanding of dendritic protein synthesis, which is very well accepted, very important in plasticity. And many investigators, including Peter Hollenbeck here, have identified axonal mRNAs, including important cytoskeletal mRNAs. Of course, just identifying them doesn't show that they're doing anything, but Peter's lab has demonstrated translation of those and in growing chick sympathetic axons, and showing that they do constitute about 5 to 10% of the total neuronal RNA. And again, that's a case of growing axons. So to show that this is really functional, one needs to show that there are protein synthesis dependent activities of growth hormones. And the example I'll show you is, for example, from turning. So Campbell and Holt in 2001 showed that applying cues elicited protein synthesis within only 10 minutes in xanthus axons. They also found rapid phosphorylation of translation initiation factors. But more importantly, when they inhibited protein synthesis, this stopped the response to these cues, the turning responses. But they also saw the same results if they cut the axon and separated the growth cone from the cell body. So the growth cone in this case clearly was acting autonomously, and there have been several other similar demonstrations since. So now I'll switch to our collaborators. This is, some of you know Jose Sotelo. His nickname is Boli, which is short for Bolito. Yes? And collapse, yes. Turning and collapse. In response to some cues, the growth cone will collapse. Yes, and please interrupt with any questions. Just to introduce you to our collaborators and show you a little bit of Montana scenery. This is Jose Sotelo. This is my wife. We're out on the hike when he visited us. This shows you the kind of views you get from our mountain ranges. Very similar to here. About as dry, too. Even back, so Boli has been working on this for 30 years now. So even back in 1982, he showed that you cut rat sciatic nerves and applied tritiated uridine to label RNA or tritiated amino acids to label proteins on one side, only to the right ones that were severed, not to the left. Those were controls. And what they showed was that label was only found in the right chunk, showing that the cell body wouldn't be the source of these, otherwise it wouldn't be able to see it in the left chunk as well. But they also saw a labeling gradient for both RNA and protein. They saw a labeling gradient from the Schwann cell into the axoplasm. So this was very suggestive, although it didn't conclusively show that newly synthesized material was being transferred from the Schwann cells to the axon stage. So he's been beating his head against this problem for 30 years. And a couple of other important aspects of this are these structures called PARPs, or periaxoplasmic axonal ribosomal plaques. Now, Zelena, back in the early 70s, identified units of just a few ribosomes near nodes of Ranvier and the Schmidt-Lanthaman incisors, and also near Schwann cell nuclei. And that, keep that in mind as I show you our data on RNA transfer. Ed Koenig and his co-workers, which included the Satellos, also found these in the Mockner axon of goldfish, a very big axon easy to work with. I didn't finish that. They later found it in the mammalian axons as well, the PNS. And the Satellos, especially the younger Satello, has learned this trick from Ed Koenig. This is, it's just wonderful craftsmanship. They can pull axons out of the myelin sheet so that you can do immunohistochemistry on the axons without permeabilizing them. You can do in situ hybridization. And the person who went to Ed Koenig's lab to learn this was, his name is also Jose Sotelo Silveira, and his nickname is Koja, and he's the third Sotelo, because the first one was a junior. This shows him visiting me in Montana, taking him skiing, and I like to show this whenever I go to Brazil to one of our courses that we teach. So they established that many transcripts are there. They found myosin 5A as well as myosin 5A message. But a very, this came out very fuzzy, I apologize for this. A very important paper that was published in 2008 was published by Felipe Court and his collaborators, including Jan Van Menen in Calgary. What they showed was that they cut the sciatic nerve of a WLDS mutant mouse, and these axons don't degenerate. That's the mutant phenotype. And then they infected with a lentivirus expressing an EGFP tag ribosomal protein. They could see labeled ribosomal protein in the axons. And it's punctated up, it looks like it represents bunches of ribosomes. So they've shown this for ribosomes. I'll show you our data for RNA, but the point I want to be sure to make right now is that they both could be going together, they could be going separately. If they're going together, we don't know which one is the briefcase and which one is the handle. So we've just established that both of them can get in.
So this is after injury, right?
Yes, this is after either a crush or a cut. I think it works both ways. Go ahead. Well, for the RNA, we don't see much labeling. We see very little labeling. So I have to say that up front, but we're not sure this operates in normal uninjured axons, what I'm going to show you. So now I've given you my amateurish view of the neurocell biology, and now I'm starting a different thread, because usually I give this talk starting with myosin 5, which was my passion for 20 years, but now I'll start bringing the myosin 5 in. So we're switching from axonal protein synthesis as a phenomenon by itself to looking at RNA transport and tethering in fibroblasts. And this is intracellularly because this is what led us to this place. So as I told you, I gave you a little bit of this. What happened? Oh, I see. I'm running on battery. So any questions so far while we wait for the power to come on? Yes. No, it's mostly people who have been looking at the axon. Yes. I'm sorry? Oh, I'm sorry. Yes, the Schwann cells myelinate the axon. So they're glial cells. They do this fantastic inside spiral to wrap the axon very tightly. So in the picture I showed, I'm going to do this on the board. So in all of these longitudinal sections for home mouse, you'll see the Schwann cells often labeled, and then the axons in here. Okay. Sure. So you have the axon, but in all of these, you'll always see a very dark lesion, or you should see a very dark lesion between this region and here. It should always be black because it's very, very tightly wound myelin. There's very little space for anything to get in or out. It's just there primarily as a lipid-based insulator. I don't know. I'm trying to get it published before someone gets to figure that out. Oh, yes. Again, I want to make clear that I use this as a supplement to fast axonal transport of the ribosomes or slow axonal, all axonal transport. This would be a supplement. And I think one evolutionary scenario that you can think of is the axon doesn't need this in an adult organism when it's functioning normally. Where it would help, where you can see a huge difference, would be during growth, because if you could feed that axon a lot of beta-actin mRNAs to drive the growth hormone better, to provide more action for the growth hormone, that will help, or after you. Yes?
Just a little added note. Another body of work that has looked at this question is an interesting one that Ray Lassig did back in the 70s, early 80s. And what happens is that it turns out that if you look at stress response, what normally occurs is that cells that are stressed by heat shock or whatever will shut down most protein synthesis and upregulate chaperones mostly. And what you find in the nervous system is that neurons have very, very weak heat shock response. And that makes some sense, because if you have a heat shock response and you need some help out in the axon, you can't synthesize more and you're down. But the glia had extremely robust heat shock response, and he was able to show that there was a substantial amount of chaperones actually transferred into the axons, in both the giant axon in a depletion model and evidence in
Yes.
So it's a part and parcel of the same sort of pathway.
So if you think of it evolutionarily, you could get a lot of selective value if you can help an axon grow more quickly or recover more quickly. You could be bigger, you could be faster. Or you could grow more, reach adulthood more quickly. So why had Myosin V with this, other than the fact that I met this was held in the zone of meetings and courses there. Of course, it's a lot like this. So we have this diluted co-color, but we have a neurological phenotype, number one. We have a human disease that has the same phenotype. We see some morphological abnormalities in the CNS. People have shown that dendritic spines and Purkinje cells are changed in these, but no one has ever connected this explicitly with the pseudo-seizure phenotype in the death. And there's also a defect in long-term depression, but not in long-term potentiation. It's also ubiquitously expressed before you can see any phenotype in non-mucus. As I said, that it's lethal, but it's not as lethal as it should be. But connecting it with RNA transfers that in neurons is found in MRP complexes with a lot of the usual suspects. And that was started in 2002, and there have been a couple of papers since implicating it. So here is purely circumstantial evidence. We have both the myosin V protein and its mRNA present in these periaxoplasmic ribosomal plaques assayed by pulling them out from the myelin sheath and doing C2 hybridization on the axoplasm. Intracellular tubular structures connect one cell to another, and those contain myosin V and actin. Axonal ribosomes have been observed, in EM, where you can see just about anything, and I'm not inclined to believe them necessarily, suggesting glial origin, at least in terms of the membrane topology. Members of the myosin of this family function mRNA transported in yeast. Yeast have cables, and these things move RNAs in yeast. And then, of course, it's a prerequisite for cell-to-cell transport of an organelle from the lantosome to show that it's caused the skin and the cochlear phenotype. And what I'll show you now is that it functions in intracellular distribution of total RNA and specific RNAs. And I'll keep going, but I could keep going, but I don't think I'll get to the data. So Aldo Cagliari from the Sokola lab spent some time in my lab because we have these lethal mice that don't have the myosin V protein. And he used a total nucleic acid stain called cytoRNA select. And we saw a really fascinating difference between the mutant and wild type cells. So if you look at the wild type cells, the nuclei are very clearly defined. And if you look at these mutant cells, in some of them, it's very hard to distinguish the edge of the nucleus. And this holds up over a very long, many samples, many repetitions. So there appears to be some very kind of vague defect in transcript localization, not necessarily transport. And here I get to go back to what I did as a postdoc 25 years ago. Because it turns out that if you want to think about this in neurons, brain has a special splicing pattern in this small region of the myosin V tail. It has this tiny little exon called exon, I call it exon V. And it goes 3 amino acids, and it's required for interaction of the thymine light chain 2. So it's this kind of dimerization, you could do it as a dimerization potentiator or something. So it's originally isolated. This is one of these confusing things for people going back in the papers. It was isolated as a thymine light chain, and it turns out to interact with a whole lot of other things. So it's required for that interaction. Now, Veronica Salerno, a Brazilian scientist who did a sabbatical in my lab, and Bill Province, a postdoc in my lab, did an experiment in which, of course, you see a mutant phenotype, the first thing you want to do if you're a geneticist is rescue it. So they transfected with full-length myosin 5A. They did bromourine labeling of RNA. This has a little different contrast because it's post-labeled, but here you can see the RNA is getting out. The nucleus is not labeled heavily with the bromourine, whereas the untransfected cells do have heavy nuclear labels. Then when we made when we used a construct that lacked this brain-specific exon V and would not interact with this LCA, which has also been implicated in some RNA transport, when they tried that, they did not rescue the phenotype. So here you see the BRU. You've got heavy labeling of the nucleus, and the transfection didn't help it. So, as I mentioned in the other talk, when you use these myosins, you overexpress the tail fragment, right, to get a dominant negative phenotype, which should disengage the myosin from its target. So in this case, Veronica transfected the wild type cells with this tail fragment, again containing this exon V, labeled with BRU, and she saw no effect. So the experiment's a huge failure. But luckily, at the same time, Veronica had some mutant cells in the incubator, and she had done those at the same time, simultaneously. So she did mutant cells, again with just the tail fragment, no head, no motor domain, and boom, it rescues the phenotype. This really, I had to think about this for a long time. Anyone have any? Why would the tail fragment or lacking the motor domain rescue this phenotype? Any explanations? It's like people didn't have tea and coffee. An adapter? Okay, anyone else? So that's the explanation we came up with. It was an estimate. Maybe the particle is actually being transported by a more important motor like kinesis. And it's just a passenger, and that's enough. And again, you're in an overexpression situation here. You even get some little, what I would call loogies of myosin 5A tail. So, maybe it's grabbing things at the periphery, and you don't really need that in this case when you overexpress it so much. I'm sorry, this is the failure. This is the rescue here. If you look at the BRU here, you can see that the nuclei are labeled, but there's no nuclear label on the transfected cell. And here we quantified it just to show that here's the full length we could rescue, lacking exon V we didn't rescue. With the tail, which I call brain tail or V tail, we We did rescue it with the tail lacking the exon B, we did not. And it didn't make much of a difference in wild type cells. So again, to show this to you in a little more, let's just keep going. That's so pleasant. So then the next question is, okay, is moving RNAs around the cell, we're labeling bulk RNA here. We're really more interested in messenger RNAs. So which mRNAs is it transportable? And of course, the first one you want to try, and makes a little sense evolutionarily in terms of growth clone migration, would be beta actin. And it's also the RNA about which the most is known about its transport from the cytoplasm and fibroblasts, primarily from Ralph Singer's work. So it turns out that when Alder did this in situ hybridization, when he looked at the wild type, again, these are fibroblasts, he saw a really nice labeling of the leading edge, which is where you're supposed to see beta actin mRNA. But when he looked at the mutant cells, he saw things kind of stopped up in these puncta near the nucleus, and no leading edge labeling at all. So this is involved in beta actin mRNA transport, and that made us feel much more confident about the whole thing. So just to review what I told you, we know that this exon B required, which then suggests that this, what was called DLC2 and is now generally called LC8 is involved. The RNA, here's the package, and this head domain is not required for this rescue of fibroblasts. Yes? No, I don't have any data. We didn't find a control RNA that gave us. So we were labeling bulk RNA, and I should add, sometimes to get a better nuclear cytoplasmic difference, we were doing pulse chase labeling as well. So it appears that, these cells appear to be fine from everything you can see on the microscope. So it appears to be more temporal, this disruption.
The maximum very you see the distributed mRNA matching.
Right. Well, it could be not getting out of the nucleus, could be part of it. So Roy Larson's lab has suggested nuclear roles for myosin V, even in splicing. I haven't seen any evidence of that yet, but I'm not doing this one. So I don't know whether it's the nuclear cytoplasmic transition or once to the cytoplasm. So now let's go back to the system. And Bombay devised it took him 30 years, but he came up with a really, I think, airtight experiment to demonstrate that RNA was being transferred. So the experiment starts with cutting the sciatic nerve of a rat. The next day, you then cut proximally to that, again, more towards the back, so you're taking the proximal segment of that sciatic nerve, and you drop that whole segment into medium-containing bromouridine to label RNA. So it all came together from the things that people had done in my lab. And then, so you wash them and fix them, then you detect RNA with an anti-BRDU antibody, the first one. And then we counterstain with antibody against ribosomes or with colloidin to see F actin, because we suspected that this RNA would be associated with that. And then it's very hard to run this, but you tease the fibers of that transected end so you can see single fibers. So with the rat, we can do homos. So as you had asked about the Schwann cells, here's a diagram of a Schwann cell. So here we have the nucleus in the outer wrap. So this is a longitudinal section. So this Schwann cell has wrapped from the outside to the inside, and these are actually way too wide. They should be, membrane right next to each other. So you have this black region, and then the axon would be in here. And is everyone familiar with the nodes of Bombay? Okay, so here you have a node. So if you think of what's the quickest way for RNA to get from the nucleus of the Schwann cell to the node of Bombay, and the answer to the axon, and the answer is this outer wrap, these actually should be closer together, is the shortest route to the axon, and that will be at the node of rendezvous. Okay? And indeed, when they looked at these fused fibers by confocal microscopy, this is indeed what they saw. So the green in this case is Roma. So here you have that outer wrap of the Schwann cells. It looks very much thinner than we've drawn it here. And this black area here is then the compacted minor. And here at the node of rendezvous, you have this beautiful punctate labeling of RNA. You see the actin in the axon. There's some colocalization, not much. But it diffuses out from that node. So that fits perfectly with this diagram, which of course was made before we did the experiment. Does anyone believe that?
So this is normal, this is not.
These are injured axons. Right, this is very close to the injured end.
If you do the same experiment in normal axon,
You see very little labeling. I'll show you the low power view and you'll see the gradient of labeling. It goes both ways. You can't really tell if this is proximal or distal. But I'll show you in a minute, I'll show you a series of distal to proximal series. So you could say, well, how do you know that's really in the middle of the fiber, that it's really the axon? Well, you can do Z-stacks and show that the labeling here is right smack dab in the middle of the fiber. You also see some labeling of bands of Charcot seem to be filled with RNA, and that's even more prominent in the mouse. So again, just to show you the experiment. Now, if you look at the cut end, so this is the cut end of the nerve. Here you see these fibers, and you can see a gradient of BRU labeling. It's highest at the injured end. And here they stained with an anti-ribosomal antiserum, and you see a lot of accumulation of ribosomes at the site of injury. And again, the 2008 paper from collegiate court suggested that those had been transferred in as well. But what heartened us was that there wasn't complete overlap here. So it justified, I think, looking at the arms separately. So here's the series that goes about. So here's actually the injured end of one particular fiber. You see this huge amount of labeling here. Here's a Schwann cell nucleus that's labeled very well. And if we go back, so we're going again proximally. Here's a node of rendezvous that's labeled very nice. The little white outline has been added to show you where the axon is. Here's another Schwann cell nucleus, another node of rendezvous, and if you go back far enough, the node stops being labeled. Yes? I'm sorry, no, we have not. Yes? The ribosomes? Yes, so that's the red here. Before the cut. No, we don't really see anything in that injury.
So then that implies the ribosomes are, since it's already cut, that means it's more synthesized.
Yes, that would be the implication from the collegiate court paper.
can you exclude the transport from the cell body of the medicine?
So, Scott may disagree, but there's only a very tiny opening in their paper. So what they did was they applied lentivirus to the injured end. And so it would have to be very, very fast axonal transport for that virus to work its way out to the cell body and then ribosomes could come back down. So they did theirs in the mouse. This one is airtight because the cell bodies are in the dead rat. But I think it's pretty much impossible that their lentivirus could have labeled ribosomes in the cell body that were negative, given the time. So here's a better generation diagram where I actually have the myelin is now compacted. Yes, I'm sorry.
Even that chemical assembly is from every element of the ribosomes actually is just a small
This is they use one ribosomal subunit tag of GFP. I forget what's one. I don't remember off the top of my head.
But you can simultaneously label small signals and maximum to the
No one has done that yet.
So would you see it in every axon that you look at with this method? You know, one problem with the predictive paper I have everybody keeps saying the predictive, and I look at it and I see the error about that. And I really don't see the body of data to work on an existing well-established model. That's the problem that I think we still have in this one, but pretty much you see it in every axon.
No, it's heterogeneous. So I think when you enter them like this, probably some of them you kill, and I'd say about 60 to 75% of the axons you see good labeling like this. Yes?
You might want to repeat the question in the future because
I'm sorry.
There's a lot of reverberation.
Yes. So here, in addition to making the ions better compacted, as I learned more neuroanatomy, I also added the Schmidt-Lantern inside. So they're a solution to the problem of the cell taking care of all these very tight wraps of myelin. If you would unwrap the cell, they would be like little tubes on top of the Schwann cell. So these are another way potentially for RNA to get from the Schwann cell to the axon, and that was also suggested by Jeffrey Twist in the review. And indeed, that's also what we see. So here's your Schwann cell, your outer wrap. Here are what appear to be Schmidt-Lantern incisors, labeled with BRU. Again, this is an actin counter stain. Here's some more, very nice incisors. We also do see co-localization with the MAG marker for these incisors. Yes? We don't make them. They're an anatomical feature. So, I told you about this. It started on Myosin V. I went back to this really nice, elegant experiment that I think overcomes a fairly large hurdle in the field. Why am I talking to you about Myosin 5A? Well, one thing is, if you do co-labeling with this BRU and the Myosin 5A, you see a lot of co-localization. So much that you can even get FRET. And I'm not sure how much that means, whether it's just all packed in, but there's clearly suggestive data for co-localization. Also, we know that actin is required for this because the truncal A treatment, if we put it in that labeling dish, that abolishes the axonal signal while leaving it in this outer wrap of Schwann cells. So actin is required and Myosin 5A is associated. So what I did at this point was I tried to take the experiment that was done on the large Uruguayan rats and adapt it to my homozygous mutant mice, which die at 3 weeks of age and have a phenotype at about 12 days of age. So we're talking about doing this on 10-day-old mice. So there are a lot of differences, especially in my eyesight and my back after doing this. But so doing this, yes?
So, it's a huge amount. Is the green thing
No, the green thing is newly synthesized.
is it all of them coming from this?
Yeah, that's unless that's the only place they can be.
Actually, John, I was wondering, have you done a control where you just put like silence mind or something in that block?
We've done alpha and in that block, most of it. Yeah. So clearly some of it is multiple space. Yes. Yeah, that's been kind of hard because you need enough to see it. Most of these are with 6-hour labeling. You can see it back to 2, but then it starts going away, and it's not the kind of thing that lends itself to a careful kinetic analysis. But we've done one in response to the reviewers. It has very wide errors. So it comes up from 2 hours is the first time you can see it in the racks and the label.
No, is it injury or the more injury you are, is more annoying to transfer that kind of solution exists?
I don't see a way to do very partial injuries. I mean, I've done, in the mouse model, I've done crush versus cut, and I've seen exactly the same thing. I've seen the difference. Is that a question? Can we do less injuries? We haven't done anything so far.
And do you see the same kind of package or
Near the injury site. So there's I showed you this. This is a series of one fiber coming from the injured tip proximal. Each one of these panels represents a proximal signal is more proximal. And sometimes, of course, it'll be the nucleus of the Schwann cell. They should be labeled very heavily, right? If this is correct. And then sometimes you see it more prominently. Okay? Yes?
Have you compared the axons in the proximal stump versus the distal axon?
Not really. We've kind of wanted to avoid the distal one. We're having enough trouble selling this.
Well, this is actually all distal. It's a distal feature.
Yeah, it's the distal. This is the injury site.
I'm talking about actually in the surviving this axon segments that are here. As opposed to the ones that are attached to the cell body. Do you see any difference between this phenomenon, different nodes of Bombay and certain the proximal stump versus this stump?
This is the proximal stump. I'm sorry.
No, it's not because you've removed the cell bodies.
Yes.
Okay. So I'm talking about the one. See, this doesn't degenerate.
Right.
If it's attached to the cell bodies, it can degenerate.
Correct.
So
have I done it with cell bodies attached?
Yes.
Yes, I've done that in the mouse. So in the mouse, the mice are small enough and I can afford the bromourethane. So in the mouse, instead of taking them out, I also would just inject the mouse IP with bromourethane and saw exactly the same thing. Thanks for asking. Okay. So this is a little washed out with this projector, but here's the experiment. So with wild type sciatic I had to do it differently because these fibers are so small, and I'm much less adept than the people in the Sotelo that I mounted them and I sectioned them longitudinally. So you're seeing sections that go, there's going to be some waves going in and out, but the difference is still clear enough. You see the axons of the wild type sciatic nerve are just filled with BRU. We also see really extensive labeling as the hollow here. However, when we look in the mutant mouse, all of the fibers are hollow. Where you see things that look like labeling, that's where the section is going along the edge of the fiber. And just as with the velocity data I showed you back in Mumbai, I got a course summer undergraduate students to quantitate all of these, and if you look at the mutants, you can see the hollowness in terms of the normalized signal of BRU versus the wild type fibers, which are clearly labeled. So here, clearly now, myosin 5A is necessary for this to happen. Yes? We don't have any myosin 5 in the mice. The problem could be that I did that personally, but I screened 700 ES clones and couldn't knock it out. But consistent with that, when I went and looked in the repositories, they didn't have knockouts either. But no, that's clearly something you want to start doing is to think about myosin compensating. But there doesn't seem to be any compensation here. But that was the case for the mitochondrial story that I was talking about, the failure to see the difference. So it's required, so the way I'm thinking about it, and in terms of packaging this for publication, I didn't want to get this out before people maybe show any more of the mechanism. Because after this point, after showing that actin and myosin 5A are required, then the whole thing gets a lot easier. How does this work? Can anyone offer any hypotheses? How would you transfer RNA from one cell to another? Pardon? Endocytosis, gap junctions, Bill? Cytoplasmic bridges. Anyone else? What about exosomes? The Sapelo lab has some preliminary data suggesting that in rats that they're seeing some co-localization with an exosomal marker. But of course, then we're layering one thing that a lot of people don't believe in on top of another thing that a lot of people don't believe in. Anyone else? So remember that myosin V is required for another intracellular transport from the melanosomes to the keratinocytes. So my view of it is very simple at this point, is that myosin V is just retaining things in the staging area for transfer by some mechanism X. So where we're going with this, where you have been to, is that we're looking for myosin V interacting mRNAs. So if you think about the evolution of this, the way it would evolve is some random accidental mutant that does this. And so you would think that it would be a bulk transport event. You wouldn't think that things would be specifically addressed to transport. Maybe if you sent all the Schwann cell RNAs in and you have enough beta-actin mRNA to make a difference, that would be enough to select for this. And sure enough, we've done it two different ways. Koja, the youngest fellow, he immunoprecipitated rat RNPs with an anti-myosin V antibody and a heterologous control. Whereas I took my tail constructs and transfected them into neurofibrillary fibroblasts. So there's a problem reconciling the data because you have completely different transcription profiles. But what we ended up doing was a Boolean analysis. In other words, if we started with what we pulled down from the fibroblasts and look for things that were threefold higher, and you go look in the RNPs, and in about 145 out of 150 cases, there's concordance there. So the Boolean analysis makes it look like this is really looking at interacting mRNAs. So there are hundreds. Then when I went on sabbatical starting in 2009, and my project in Jim Switch's lab was to learn biochemistry at a very late age. And what I did was I put, again, those two tail constructs that I showed you, with and without exon V. It was I thought it was a very elegant experiment. And we found 1,300 proteins that interacted with the myosin V tail. And if you go back to what I told you before, if it was present in the ancestral eukaryotes, had a chance to pick up a lot of different functions. But there were about 100 that differentially bound with the exon V containing tail, and that contains many RNA binding proteins. And I'll show you just one of them that looks very promising. And that's HNRNP-A2B1, which co-localizes pretty well. So that's the blue in this case. And here's the axon state, so you see a lot of the staining in the axon. But that's not very good evidence about itself. We also know that it interacts directly in vitro. So finally, what's going on now, even though I'm here in India, I still have access to the animal work back in Montana, and we're doing both transcripts in collaboration with Jeff Mercer of the Weizmann Institute, and ribosomal tagging, where there's a ribotag mouse where you can use pre-lock technology to specifically label ribosomes in one tissue. So if we tag ribosomes in Schwann cells, do we see them in the axon, for example? Do we see them after injury? I would hope so, but this also lets us get to the point where we can start to ask the question, does this occur in normal axons that aren't growing or responding to injury? Because I think in the transgenic mice, we can let things accumulate long enough to hopefully see them. So I'll stop there and acknowledge Jose Sotelo and Aldo Lucia and Alejandra in his lab, Koja, who's at both institutions in the Uruguay, Bill Province, Veronica Salerno from Rio de Janeiro, Colleen in my lab, and Jim Spudich, and this was funded by NIH Program International Collaboration Grant. And I'll stop there and take any more questions you have. Thanks. Yes. So the question is this important for healing of the wound? So the way I think about it is, if you look at this again evolutionarily, the axon will have ways to stop the damage basically and just scar off and stop everything versus regeneration. And the appeal of this, the thing that the ground reviewers like about this is that it allows a way to potentially control the proteins made by the axon, potentially, potentiate recovery of function. So, for example, you can put the cut ends in the collagen sleeve, and that promotes it. You can put it in Schwann cells, that makes it work better. And you can potentially put skin-derived stem cells from the patient into one of those collagen sleeves, and if you can tell them what transcripts to make and get that tag properties go into the axon, that would be very valuable. Yes?
I think some mechanism like that in invertebrates where non-myelininated exons.
Is there a mechanism like that in invertebrates? I don't know. But Scott just said we had some evidence that we had transferring in the squid.
The can go back a bit like because we had if you go back, folks had the main experiment where you demonstrated the transfer.
The rat experiment?
Yeah. Yeah.
This?
So can you explain exactly what you're doing to you think that
Sure. So you start the experiment by transecting the sciatic nerve. You staple it up or glue the incision, and then the next day you excise, you make approximate to that initial injury. You take out that segment, you incubate it with bromouridine. Or as I noted with Scott's question, in the mouse I could also inject DRU and that works the same, so that it's not an artifact of the culture system. And then you just do your analyses on that fragment after you fix it and piece it. Backward, away, forward. Forward. Here?
so all the angles in here
Is newly synthesized RNA. Yes, it's a lot. And I can tell you, so again, if this is a 6-hour incubation, you can go back to 2 hours to still see it. But in the mouse, when I did this, I couldn't make the time shorter. I couldn't do multiple mice and have it say, less than 20 minutes. Even in 20 minutes in the mouse, the entire axon was labeled. If you notice, there was no gradient like this in the mouse.
if you need the transfer, then you might be able to show a sequence of events where it is first in only the Schwann cell, and then only in the midline, and then only basically that would be
Yeah, I think that's the next thing to do, but I think first we have to convince people that this is real. But if we had done the experiment in the mouse, we never would have had any handle. We never would have seen this gradient. These RNAs belong to only Schwann cells. Well, we don't know.
What are the chances that those things with the factors are coming from the
Right, the cell body is in the dead rat and can't be labeled with DRU. So they have to come from the Schwann cells, or there could be some other cell type, but look at how the Schwann cells are labeled. It's even more unlikely that it would come from any other cell type. Yes? Is there any evidence that it works in the reverse direction? None that I know of that really well.
From your exam country, cargoes of Myosin V can stage from
Right, so this LCA binding, it just seems to potentiate the dimerization of the quote-unquote that's on either side. So what I would suspect is that binding changes, if it's interacting with 1,300 different proteins, maybe that changes it, just a little bit to change the spectrum of proteins with which it interacts. LCA has also been implicated itself in some RNA transport, but I'm not sure I believe those because there's very little follow-up on it. Yes. Right. Yes, possibly. There's a lot of heterogeneity. I could show you. I could show you. Oh, again.
Do we have any culture system, culture system where we have concept and exam together going and they might shape? So do you think so in the
Yes, we do, but I would say, and I think I would hope everyone, if I did those experiments in that system, no one would believe it.
Yeah, I don't know.
Right, then you can go back to the co-culture system. And then we can, so it's easy to envision doing a co-culture system where the swan cells are from the mutant mouse and the axons are the neurons are from the wild type mouse and the complex as well.
And also, if we instead of doing like that, Myosin V, Myosin V.
Right. Although I like mutants because they're easy. But yes, yeah, I thought of those, and also I thought of trying to do this with standard precursor cells, so you would just implant those instead, roll those up and freeze those down and implant those at the site of the tree. See what we get.
Okay. Yeah, go ahead.
Okay, last question.
Is there the scope of RNA regulation in this?
Is there any scope of RNA regulation? Not that I know of. But I'm sure that it
It's good to see that there's one thing we did doing after the long bus rides. So we'll take a break now. Tea will be served in about 5 minutes or so. So please have your tea and come back here a little before